Hello everyone. Today I discuss about the Mendelian genetics and its extension. It is the part of core syllabus of plant science, that is botany. So first of all, we have to know about the genetics. Actually, genetics is a study of the hereditary, and heredity is a biological process on which the parental or parents passes their character from one generation to another generation, that each parents passes their character to the offspring or their children, and every children or every child or offspring inherits character from both their biological parents, and these characters are regulated by a special uh, unit of the DNA, we know that, and this is the gene. And this gene or this unit specific sequence express itself under different environmental condition as known as phenotypic expression. Let's we know about we have to know about some terms of genetics, and these terms are maybe gene, chromosome, linkage, trait, character, and there are several terms related to the uh, genetics. I just discussed some of them. First one, we know that every cell has a cell brain, which is known as nucleus in eukaryotic cell, and in nucleus huge chromatin fiber are present within the nucleus, which is known as chromonema, and these chromonema under condensed condition form a special rod-shaped structure which is known as chromosome. And each chromosome contains several units, and these units are the nitrogen base sequence made the genes. Next, the allele. We know that in chromosome, the character regulating factor, that is gene, present in a specific location which is known as locus. That is, the location of the gene is known as locus. And these genes, each and every gene has an alternative form of property. This alternative form, that is the pair of factor, occupy the same position, that is the locus, in the chromosome, and these are known as allele. That is, the alternative form of a gene is called allele, and these alleles, when are present in a single locus, this is known as gene. That is a section of DNA which codes for the formation of a protein controlling a specific characteristics of the organism. There are different nucleus: haploid nucleus or diploid nucleus. Haploid nucleus, that is single set of unpaired chromosome, and diploid nucleus, that is pair of chromosome in somatic cell. Somatic, that is body cell. The diploid number is 46. Next, the trait. The relative characters which are expressed, that is, which have the phenotypic expression, is called trait, and these traits are regulated by a specific genetic makeup which is known as genotype. Each and every genotype, I just discussed that every gene has an alternative form. That is, the genotype has an alternative form, that is the either in form of capital T small t, or in form of capital T, or in form of small t, where capital T and small t are the alleles of a gene. That is, capital T and small t, these are the alternative form of an single gene. Next, the phenotype. I discussed that the genotype which is express its character under different environmental condition, this is known as phenotype. That is the morphological expression of the character in different environmental condition is called phenotype. The characteristics visible in an organism controlled by the genotype, that is a tall plant or a dwarf plant. Tall plant is regulated by the capital T or capital T small t, whereas dwarf plant is regulated by the small t small t. Next, homozygous. We know that homo means same, zygous means pair. Having a pair of identical alleles controlling the same character, or that is same type of identical type of allele, as for example capital T, this is the homozygous. Again small t, this is also homozygous, where t is equal to tall. The organism will be pure breeding for that characteristic. Next, the heterozygous. We know that hetero means different and zygous means pair of. Having a pair of dissimilar alleles for a characteristics is called heterozygous. As for example, capital T small t is a heterozygous allele. Next, the dominant and recessive. The character which is express itself when the alleles are present in the heterozygous condition, this is called dominant allele. Suppose the capital T and small t are present together when only a small capital T is express itself, that is the small t cannot express itself. Here the capital T is the dominant allele. That is, the capital T express its phenotypic expression. Recessive allele, that means in capital T small t or in heterozygous condition, when the capital T express itself but small t cannot express itself, it is called recessive allele. So dominant and recessive, these are the two forms in terms of expressivity. Other terms also there in genetics: hybrid, hybrid, lethal allele, pseudogene. So I discuss these in later case. Next, come to what are alleles. If you see that this is the same two types and this is the two different types. If we look at this board, then there are two forms, capital A and small a, out of which the capital A and capital A small a and the small a small a, of which in these three cases, in these three cases, capital A only express itself, whereas the small a express itself in homozygous condition. So from this view, we can clear our concept about dominant and recessive. So these are the alleles. The recessive alleles are symbolized by the lowercase letter and the dominant alleles are symbolized by the uppercase letter. Next, come to the Mendelism. As we know that Gregor Johann Mendel is the father of genetics. He is the pathfinder of the genetics. And especially Mendel gives some principles of the genetics, although his principles are not previously recognized. After his death, after some years, finally his principles are recognized by the scientific community. The Mendel was an Moravian monk and biologist who established the laws that are the foundation of classical genetics. We know that all the practical cases, all the practical purpose are completely depends on the classical part. So genetics is not an exception. All the application of the genetics completely depend on the classical genetics, that is Mendelian genetics. In 1865, Mendel's discoveries, where all the laws are unnoticed for some years, finally in 900, that is 16 years after his death, Mendel's paper was rediscovered independently by three different scientists. With the rediscovery of Mendel's work, he came to be recognized by the scientific community, and his law, three special laws are recognized by the community. I just discuss about this law briefly. The Mendelian genetics is a type of biological inheritance that follows the principles originally proposed by the Gregor Johann Mendel in 1865 and 1866. And there are three special laws: law of dominance, law of segregation, and law of independent assortment. In case of law of dominance, when heterozygotic condition is present, only one allele expresses itself. This is known as dominant allele, and which cannot express itself is called recessive allele. And the property of the expression of dominant allele is called dominancy. So one allele of a pair of alleles controls a character, usually express itself and prevents the expression of the other. The allele which is expressed in a hybrid condition, that is heterozygote condition, is called dominant allele. And whereas the unexpressed one is called recessive allele, and this law is known as law of dominance. If we think about that, if we cross a tall plant with the dwarf plant, then in F1 generation, the tall character is expressed, although the genotype of this plant is heterozygous. That is, the dominant allele expressed in heterozygote condition, and this is the law of dominance. Next, the law of segregation. During the formation of gamete, although the different form of allele, that is capital T and small t, are aggregate, are mixed in the F1 generation rather than it segregate during the gamete formation. This is the law of segregation. So during the formation of gametes, the two members of a single pair of genes are never found in the same gamete, but they always segregate and pass on two different gametes. That is, during this formation, the gametes are segregated from each other. Next is the law of independent assortment. During gametogenesis, when the gametes are separate from each other, at that time the gametes reunite with each other in a specific manner. In a random case, all the gamete can pair with its opposite pair of alleles. During gametogenesis, the members of different gene pairs assort randomly or independently of one another. These are the three special law or principle of the inheritance proposed by Gregor Johann Mendel. Okay, let's think about the recessive and dominant. So if You look at this. Here, capital N and small n, where the small n is the recessive and capital N is the dominant. And if we see that the father and mother both are carrier because it has the recessive allele affection, that is recessive allele disease character, whereas dominant allele is normal. So during the inheritance, this one is normal one, this one is normal but carrier, this one is normal but carrier, but this individual is affected. So when the recessive alleles are present in homozygous condition, then the character expresses and these persons become infected or affected. When the dominant inheritance, if we see the dominant inheritance, if we cross a affected father with a normal mother, here the D is the capital D is the affected allele, and if we look at this, then both these capital T small d, that is in heterozygotic condition the character is affected, whereas in homozygous recessive condition the character is normal. So these two are the recessive versus dominant inheritance. An example, I think all of we clear our concept about the recessive and dominant inheritance. Next, come to the chromosome theory of inheritance. All we although know that the DNA is the basic unit of chromosome, the sequence of the DNA is the main unit or basic unit of the chromosome, and the genes which are present in the chromosome uh, transmitted from one generation to another generation. So the inheritance regulated by the chromosome, and in 19th century, 1902 or 1903, scientists Sutton and Boveri published a proposal about the chromosomal theory of inheritance. That is, they proposed that all the characters which have been inherited from one generation to another generation are controlled by the chromosome, are based on the chromosomal inheritance. There are some logic behind this. Suppose if I uh, uh, told about the all the haploid organism have two chromosome, that is one pair of chromosome. During the gamete formation, the chromosomes become separated or chromosome become segregated from one another, and during fusion the both gametes are fused with each other, that is the both chromosome are fused with each other, and after that characters are expressed. So these are the logic behind the chromosomal theory of inheritance. This theory states that individual genes are found at a specific location which is known as locus, and that the behavior of chromosomes during the meiosis can explain why genes are inherited according to the Mendel's law. And if we see here, this cross was performed by T.H. Morgan. That is, the experimental proof of chromosomal theory of inheritance was performed by T.H. Morgan. I repeat, T.H. Morgan performed the experimental proof of chromosomal theory in Drosophila. Scientist Morgan taken red eye male and white eye male and red eye female. The red eye female is the wild type and white eye male. And this eye color is controlled by the W plus, W plus, and this is present in the X chromosome. So at the beginning, when he crossed with the red eye female with the white eye female, then the F1 generations are all red eye, that is, females are red eye and males are red eye. After that, if we see this F1 crossing, or the first one, if you see here, the red eye female, that is the XW plus and XW, and the red white male, that is the XW and Y. Although the first chromosome, that is the W plus and W plus, W and Y chromosome, these are crossed, and as a result he has obtained these two character, one male and female. This one is the female and this one is the male. This is the red and this is the white. At the first beginning, and in F1 generation, the result is found that red eye male and red eye female, that is both the red eye character. So where the white eye character is gone, is the white eye character becomes disappear? Not. In case of F2 generation or F2 crossing, it shows that one female with XW plus W plus, one male with XW plus Y, one female with XW plus W, and another one is the white eye male. That is, all the chromosomes are transmitted or inherited from one generation to another generation, and the character, that is the gene, that is W plus and W, these are also inherited with the chromosome from one generation to another generation. So I again, if I discuss, first one is the red eye female, white eye male, these are crossed, then red eye female and red eye male is formed. After, when these are crossed, then the F1 in F1 generation, one is red eye female, red eye male, red eye female, and white eye male. That is, two females are red eye, one male is red eye and one male is white eye. That is, all the character are inherited through chromosome, and this is known as chromosomal theory of inheritance. Here it is the same, and this experiment was performed by the Morgan. Let's come to the pedigree analysis. If we would like to analyze the family tree, there are so many generations. Then if we try to analyze in the genotypic form, then it might be trouble to us. That's why a spatial analysis is developed by the scientists. This is known as pedigree analysis. All of the conclusions regarding gene action we have discussed so far have been obtained from analyzing the result of controlled cross. In some situations, we don't have the opportunity to perform controlled cross. When the tree is very length, when there are a huge number of offspring, huge number of generations are there, then a revised approach is performed, is known as pedigree analysis. In pedigree analysis, some special signs are used, that is the rectangle form, that is the male, circular round female, black color, that is the affected individual. This sign shows the mating, This is the mating. This one is the generations from one generation to another generation. This is the second generation. This type shows the identical twins. This shows the non-identical twins. So to study the inheritance of genes in humans, pedigree analysis is also useful when studying any population progeny. Data from several generations is limited. Pedigree analysis is also useful when studying species with a long generation time. A series of symbols are used to represent different aspects of a pedigree. So these are the symbol of the pedigree analysis. I just briefly uh, discuss about the pedigree analysis with an example. If you see that this one is the individual, this one is another individual, and these are the affected, and during their cross they produce both affected one and another one is the normal one. So if the gene is dominant X-linked, then all the individuals become affected. But the gene are not X-linked dominant, that is, it should be autosomal dominant. And when these are crossed, one is affected, one is normal, they also produce one is affected, one is normal. And after that, when these normals are crossed with each other, they produce all are normal. In genetically, if we analyze, this is the capital A small a, this is the capital A small a, that is all are the heterozygous, and the affected character is the autosomal dominant. And this one is autosomal dominant, this one is autosomal dominant, but this one is not, because here only recessive alleles are present. Here is also recessive alleles are present, and all the offspring which are produced, these are also autosomal dominant. So this is a small example of pedigree analysis. If you have any question regarding this, you can post in the chat box. And next part of this Mendelian genetics and its extension we discuss very soon. Thank you.